before, so I won't dwell on every aspect of this. It's a huge topic. So some of you have seen before. I want to give this from a cellular perspective, but I want to introduce the immune system and talk about some key concepts that I would likely to retain. So I'm not going to go through the learning outcomes. You can read this on your own time. There's a plan for this session. I'll talk a little bit about the organization of the bone marrow, which of course is where your immune cells originate. I'll talk about the production of blood cells, how they're destroyed and turnover, and then we'll think about the constitution of the blood. We've already looked at some blood cells. I think we've looked at some Michigan slides. We've got some really nice blood smears. And I can't emphasize enough, you want to be able to recognize circulating white blood cells, and it's not difficult. So in the self-directed learning, open up the Michigan slides and satisfy yourself that you can recognize these cells, and some you might pick up on in some of the revision sessions afterwards. We'll talk a bit about red blood cells, of course, carry oxygen, and platelets, which are important in clotting. Now, hematopoiesis is a highly dynamic process. Cells are being turned over all the time. You're creating new cells to fight infection, platelets for clotting, red blood cells to carry oxygen to tissues, and to a certain extent take carbon dioxide back to the lungs. So we're talking about billions of cells being produced. White blood cells, as well as red blood cells, produced in the billions per day. Red blood cells survive for about 120 days in culture. 120 days in the circulation before they're eventually broken down in the reticular endothelial system, which I'll talk about shortly. Platelets have a much shorter lifespan. Neutrophils last for a few days. Lymphocytes, mainly short-lived, but of course you've got your memory lymphocytes, which allow you to respond to a pathogenic challenge many years after your body has dealt with that infection for the first time. So I'll talk a bit about the bone marrow, the cells of the bone marrow, which are the hematopoietic progenitor cells. You see a band cell there, which is an early neutrophil, but we'll talk about that. I want you to look at a blood smear in the self-directed learning. You probably can't see there. You've got these are all neutrophils with a lobe nuclei, something you should be able to recognize. And then I've got the reticular endothelial system. So what organ is this here? Spleen. What does the spleen do? So it stores platelets, it turns over red cells, and unlike the lymphoid system, it filters the blood. So I'll talk about that in a little while. So majority of hematopoiesis takes place in the bone marrow in infants. It's distributed in the throughout the skeleton. With growth, it becomes more restricted. In the long bones, the yellow marrow increases in size. The red marrow, where hematopoiesis takes place, is displaced towards the ends of the long bones. But it happens. You need to turn over a lot of blood cells, produce blood cells through adult life, and so a number of different sites in adult: the pelvis, sternum, skull, ribs, and vertebrae, where hematopoiesis continues. And the pelvis is quite an important, accessible site if you want to take a sample of bone marrow. And bone marrow can be sampled in a couple of different ways. You can take a Triphion biopsy, which is an unpleasant procedure. You drill into the iliac crest, again, site of active hematopoiesis, but you take a needle, which has a sort of drill-like motion, you drill through, and you can take a core, and that maintains the integrity of the bone marrow. So you can look at the structure of the bone marrow. Here, for example, you can see the organization with the trabecular bone and the immature blood cells. But you can also take aspirates as well, normally taken from the sternum. You carefully insert a needle into the sternum and just draw off some bone marrow, where you lose the structure. But if you're looking for example, leukemic cells, which are evaded therapy, then that's one way of sampling the progenitor, the niches where these cells can hide. So bone marrow is highly organized. They've got immature red cells here and white blood cells, and they're attached to a stroma, so they're physically linked to the microenvironment, which provides signals for maturation. And then with maturation, they're then released into the bloodstream. This is an aspirate. Again, rather than a Triphion, we take a core, you just draw off some bone marrow. See some red blood cells, some myelocytes, which are immature granulocytes, and some immature lymphocytes there. But we'll talk about this in a little bit more detail as I go on. So Blood cells, of course, produced from progenitors. Of course, every cell in the body is produced from a progenitor. But of course, this is an active process continuing through adult life. So it's a tissue which is turning over. The regulation of this, of course, is quite complex. And I'm going to simplify a little bit. I'm going to put up a diagram in a second showing a whole range of cytokines. Cytokines are very complex. Under certain circumstances, they can promote immune reactions. Under other circumstances, they'll suppress it. I'm not expecting you to know what every cytokine does in every situation at this stage, but there are a few which are worth knowing about. They're important clinically as well. For example, erythropoietin, which of course you'll heard about, blood doping, drives the production of red blood cells. Thrombopoietin increases the number of thrombocytes, platelets, important in clotting. And then you've got factors, GCSF, GM-CSF, involved in the maturation of granulocytes, interleukins. Interleukin 3, for example, is involved myeloid development. Interleukin 2 is interleukin involved in lymphoid development. So we have our progenitor cell sitting at the top of the tree. So a slow cycling cell, it'll divide to produce a daughter cell and create another stem cell as well. And this is in a highly protected niche because as this is the progenitor for cells which are going to go into circulation, it's very important it's in a protected niche, protected from environmental challenge and potential mutagenic events. And there are two broad divisions, divides into lymphocytes and myeloid cells. And so the myeloid cells will eventually differentiate, produce megakaryocytes, produce platelets and red blood cells. But the rest of the cells are white blood cells or leukocytes. And if ever asked you an exam, name a circulating blood cell, do not say leukocyte. Far too generic. Specifics we want. So thinking about cells you're going to see in the circulation, so ones that don't circulate, for example, mast cells. We spoke about mast cells before being involved. They're involved in innate immunity. I'm going to talk about that. They're also involved in allergic reactions, very, very similar to basophils, but they come from a different derivation, which I believe I mentioned before. But the key circulating cells derive from myeloblasts. And I'm going to talk about these cells in a little bit more depth as we go on. The key thing, I'm sure you remember this from the exam, a cell derived from monocytes, macrophages, quite straightforward. And macrophages are an exception to the developmental rule that everything happens in the bone marrow. Macrophages differentiate from monocytes in tissues. So these are tissue-resident cells. Lymphoid cells split into B and T lymphocytes. And of course, B lymphocytes, the mature B lymphocyte, is a plasma cell. That's a cell that produces antibody. I'm going to talk about that later on. And we've got natural killer cells as well. And the biology of natural killer cells is quite complex. I'm going to talk a little about the immune system, hopefully just to prime you for Claire's talk after Easter and make sure you're comfortable with the key concepts. But what we've got here is progressive restriction in developmental fates. So we've got lymphoid myeloid cells, and the myeloid cell being a progenitor of white blood cells as well as erythrocytes, red blood cells, and megakaryocytes that form platelets. I mentioned earlier that this is controlled by hormones and it's incredibly complex, but there are a few things which are worth remembering. Interleukins, particularly interleukin 2, is important in regulating the maturation of lymphocytes. Now, natural killer cells have been omitted from this because natural killer cells are alone to themselves. Some express T cell receptors, some don't. They're involved in innate immunity, and I'm going to talk about that. But again, interleukin 2 is probably quite a key interleukin involved in maturation of lymphocytes. Now, this takes place in the bone marrow. Natural killer cells, rather B cells mature in the bone marrow. T lymphocytes mature in the thymus, certainly in the younger individuals and in older individuals, adults. T lymphocytes they differentiate and mature predominantly in the spleen. Natural killer cells, no one's really sure where they mature. Could be the thymus, could be bone marrow, but it's only over the last few years that a body of research about the biology of natural killer cells has begun to unpick their origin and function. So I mentioned earlier on, so the granulocyte macrophage colony stimulating factor is quite important across myeloid lineages, but the key factors, thrombopoietin, maturation of megakaryocytes, erythropoietin, maturation of reticulocytes into erythrocytes. I'll talk about that in a little more depth later on. And then the myeloblast, which we'll look at in bone marrow, can then give rise to a range of granulocytes, cells with granules in the cytoplasm, apart from monocytes, which are very, very, very small granules, so they're not normally considered to be granulocytes, and a range of other interleukins as well. But the key factors, thrombopoietin, erythropoietin, granulocyte and granulocyte macrophage stimulating factor, interleukins involved in these divisions of hematopoietic precursors. So peripheral blood counts, if a number of investigations center on a blood count. If you've got low hemoglobin, low red blood cells, be concerned about anemia. If loss of white blood cells, you might be concerned about neutropenia, ability to mount a response to infections. An increased white blood cell count could occur from infection. It could be leukemia or lymphoma. And again, loss of reduction in platelets can be associated with clotting disorders. So any deviations from these figures, you could be concerned about an underlying disease process. And of course, these values can vary. Different labs will have different normal ranges, but you'll always, when you're making an informed decision based on a patient's blood count, you'll always have a reference range so you can work out what's going wrong. And you can, of course, combine that with microscopy to find out which cell types are actually impacted on a blood film. This is an illustration of a therapeutic dose of red blood cells given to someone who needs a transfusion. So a number of key statistics relating to red blood cells. You've got billions of red blood cells in circulation. Again, if there's a drop in hemoglobin, you might be concerned about anemia. The mean corpuscular volume, that's the average mean size of a red blood cell. If it's too small, you might be concerned about microcytic anemia, too big, macrocytic. If you've got an anemia, but the MCV is the same, then that'd be a normocytic anemia. So of course, red blood cells are basically bags of hemoglobin. They're to carry blood, oxygenated blood from the lungs, deposit it in tissues, and then to an extent carry some of respired carbon dioxide back to the lungs, or the majority of it's returned as bicarbonate. They don't have mitochondria, they don't have organelles, and they don't use the oxygen that they carry, so most of the energy is generated by anaerobic glycolysis, which of course isn't efficient as aerobic glycolysis. And a key feature is the flexibility of the membranes. I guess you've done this before in Molecules, Genes and Disease. Again, this figure, I think I said 7.2 microns in an earlier talk, but I think we need to worry about a decimal place. They're slightly smaller than your normal circulating lymphocyte, leukocyte, biconcave discs, and they're flexible, which allows these cells to deliver oxygen through very, very narrow capillaries. And I'm sure you've seen these little rouleaux, these stacks of red blood cells. If you've got a histological section, and almost every histological section we've looked at is very well vascularized. You're only ever a few microns away from a blood vessel. In these small vessels, and here particularly is a capillary, you can see the endothelium, squamous cell endothelium, the cells stack up in these characteristic rouleaux. And when you've got a nicely prepared sample, you often see these stacks. And this is facilitated by the flexibility of the membrane. So they have to get through microvasculature, which is a narrower diameter than the size of the cell. So they've got to fold, got to bend, got to deform. And so, therefore, the red cell membrane is incredibly important. Now, you've already done ABO blood groups, I believe you mentioned it. I'm not going to talk about it again. You're going to have a whole unit on membranes next term, so I'm not going to go through this in a huge amount of depth. Other than to say that, so the glycoproteins on the surface, these are the sites of antigens which are used in ABO blood typing to ensure appropriate blood matching. Membranes are complex. They're not just a bilaminar lipid sheet. There are 50% proteins. The majority component red blood cell membrane is protein. Flexibility is mediated by these molecules here, ankyrin and spectrin. They connect with the underlying microfilaments, and they allow the deformation, the flexibility of the membrane. In sickle cell anemia, of course, you have this aggregation of hemoglobin, and that causes the cell to become rigid. And of course, there are a number of pathological consequences of this. The cells don't fold as well, so they get trapped in the microvasculature, and tissues can become ischemic. the membranes are fragile, so they turn over faster. Because they turn over faster, you end up with a decreased red blood cell count and therefore anemia, dizziness and weakness and other consequences that follow on from that. You know all about hemoglobin structure. I will mention again. Common form in adult 2 alpha 2 beta chains, carries oxygen to tissues. It's an allosteric molecule, so when there's lots of oxygen around, scoops it up. When there's a lot of carbon dioxide around, it's acidic, ischemic conditions, the oxygen is released where it's needed in tissues, and the last bits of oxygen are squeezed out by the conformational change facilitated by 2,3-DPG. So because of that, of course, you have this allosteric binding pattern. I mentioned this already, carries oxygen to tissues, carbon dioxide back to the lungs with a reversible interaction with oxygen and carbon dioxide, of course, an irreversible reaction with carbon monoxide, which is poisonous. And I believe you've talked about globin chains in the past. They protect the heme molecule. And of course, they're important in the allosteric reaction. So looking at an oxygen dissociation curve, it's a sigmoidal curve. This doesn't look particularly sigmoidal, so that when there's plenty of oxygen around, scoops up the oxygen, and then low oxygen concentrations, the oxygen is released from the molecule to tissues. I think in fetal hemoglobin, the fetus needs to scoop up as much oxygen as possible from the mother, so that the curve is shifted to the left, so it becomes saturated in lower oxygen concentrations. The number of the curve gets shifted to the right, as with an increase in carbon dioxide, and in, for example, in a sickle cell, where the ability for the molecule to carry oxygen is compromised. So you need more oxygen for it to become saturated. But again, you've covered this already. The production of red blood cells begins in the yolk sac. We have your primitive epsilon forms of hemoglobin. Alpha chains are formed from a fairly early stage. Hematopoiesis in utero takes place predominantly in liver, later in the spleen, before it moves into the bone marrow. And then the fetal hemoglobin is replaced by beta hemoglobin in adults as the requirement of the fetus to make the most of the available oxygen is no longer such an important driver post-birth. So the important thing to remember, fetal hemoglobin, higher affinity for oxygen, and sometime 3 to 6 months postnatally, there'll be a switch. And at that point, of course, any mutations in the beta chains will become revealed. So sickle cell anemia, for example, doesn't manifest itself in utero because of the presence of gamma chain, but it will become manifested quite quickly after birth. Anemia can result from a variety of different processes. Of course, you have massive blood loss through trauma, become anemic. A number of things, for example, an iron deficiency could decrease production of red blood cells, and then you have disorders such as anemias, sickle cell thalassemia, where there's an increased turnover, increased destruction of red blood cells, which isn't matched by production of new red blood cells. So there's not enough hemoglobin around to carry oxygen to the tissues with the associated effects. I think I mentioned earlier on, red blood cells basically sacs of hemoglobin, no organelles, and they get most of their energy from anaerobic glycolysis. Now, I don't expect you to remember these pathways, but these are the key energy generating pathways in red blood cells. I'm not sure if you've covered it in metabolism. So it's important that in order to deliver oxygen to tissues, that the body regulates red blood cell production. And any reduction in oxygen partial pressure will be detected in the interstitial peritubular cells of the kidney. So as with ischemia, as red blood cells fall in number, the oxygen concentration in tissues is going to fall, and these cells will detect it, and they will increase the production of erythropoietin. I spoke about erythropoietin earlier on, driving the production of red blood cells, erythrocytes from reticulocytes and the progenitors, and the myeloid precursor. Red blood cells are initially nucleated, and of course they become anucleate, release the nuclei as the red blood cells are released into the bone marrow. A very small fraction of red blood cells will still be nucleated, have been probably released slightly early, something about 1% or so of your circulating red blood cells might still be nucleated. But generally speaking, red blood cells, little flexible bags of hemoglobin. And of course, you've got a feedback loop here. So the signaling from the interstitial peritubular cells, the release of erythropoietin causes an increase in erythropoiesis. The more hemoglobin around, more oxygen being delivered to tissues, and so therefore that's picked up in these cells, and EPO production is shut down. And there's a constant homeostatic process going on there. And this diagram just shows the same thing. We'll go through that again. One of the learning outcomes is the catabolism of hemoglobin. And actually, I think it's worthwhile remembering that iron is a very precious substance. And you don't want to break a red blood cell down and then lose all the components. And actually, the red blood cells are recycled in the reticuloendothelial system, particularly in the spleen. These steps are happening here. So towards the end, 120 days in circulation, red blood cells begin to break down. So hemoglobin separates into heme and its globin chains, and the globin is recycled for its amino acids. But your body's got to handle a lot of heme. Heme breaks down, the iron is released, it's recovered by transferrin. Transferrin takes the liver, then onto the bone marrow, where it's reused for erythropoiesis. And then the heme, once you've removed the iron, essentially leaves biliverdin, and that becomes reduced to bilirubin, which is transported to the liver. So just ignore this little dashed line here. The bilirubin then is solubilized by reacting with the glucuronic acid to form bile. The bile is released from the bile duct, goes into the small intestine, and in the gut, the glucuronic acid is removed, recreating bilirubin. The bilirubin is then converted to urobilinogen, which is transported either via the bloodstream to the kidney, where it's excreted as urobilinogen, or the bilinogen is converted to stercobilin, which is brown-colored pigment, and then it's excreted. So again, I don't expect you to recapitulate this diagram. It's remembering some of the key steps in iron recovery, and excretion of bilirubin is useful to have a handle on. So moving on from red blood cells to platelets. Platelets are important in clotting. The thing is, platelets are really, really sticky. They're basically membranes containing granules which facilitate the clotting process. The reason this is shown here, just to remind you that when you're in the clinic, you don't want to leave your bags of platelets lying around. They're very sticky. They'll aggregate. They're kept shaking. And then we've got two agitated adult therapeutic doses of platelets shaking away to be given to someone who has a problem with clotting. Platelets are formed from megakaryocytes. We saw that earlier on. They derive from a myeloid precursor. And these are interesting cells. They carry on they carry on dividing. They carry on replicating their DNA without dividing. So they get larger and larger and larger. And platelets start budding off from the megakaryocytes. So the megakaryocytes themselves, you don't see these in circulation. But if you look at a blood smear, the little sort of membrane-bound sacs you can see between red blood cells will be platelets. And they're basically membranes containing granules, which I shall talk about in a second. And that's driven by thrombopoietin. So a very typical blood smear. I see your 7.2 to 7.5 micron red blood cells, the biconcave discs. And then these little speckles here, these 2 micron units are thrombocytes or platelets. If you put them on a plastic dish, look at them under scanning electron microscopy, you'll see them spreading out. It says in the bottom there, the majority of these are stored in the spleen. So they're important in plate clotting and specifically blood clotting and repair of vessel walls, which is shown in this cartoon. And I'm going to go on to explain this process. Now, you've done clotting already, I believe. I'm not going to talk about all the factors. I just want to talk about it from a slightly more cellular point of view. The important thing about a platelet, they have a membrane. It's a phospholipid-rich structure, and it provides a surface for clotting factors to aggregate and adhere. I'll talk about that. The 2 kinds of granules. One produces clotting factors. For example, von Willebrand factor is important in stabilizing fibrin clot. and a number of other factors which are involved in aggregation and also in vasoconstriction as well. If you've got a damaged endothelial wall, you want it to contract and restrict the blood supply. Again, I wouldn't worry about all of these factors in minute detail. So when you've got damage to the endothelium, it will reveal underlying connective tissue. Damaged endothelium also releases von Willebrand's factor, and the platelets recognize via the cell surface glycoproteins exposed moieties on the collagen which has been revealed as a consequence of damage. And this forms an initial loose platelet plug. And that's your primary aggregation or primary hemostasis. But the clot has to stabilize, and this is when the cascade begins to happen. And again, I'm going to simplify this, but degranulation of the platelets recruits more platelets, and of course you have this cascade. The platelets also release ADP, and ADP provokes a conformational change which reveals other glycoproteins, particularly glycoprotein 2B and 3A. And these now make the platelets more sticky, and they encourage aggregation. So the plug increases in size. Then fibrinogen is recruited. It's converted to fibrin, begins to polymerize and form a clot. Now, of course, this is the end point of your various factors, which I'm not going to talk about, but they impinge upon the activation of thrombin and ultimately the polymerization of fibrin. But what you end up with is platelets, which perform this surface for recruitment and polymerization. They trap red blood cells to make the clot, and it's encapsulated or supported by this pretty fibrin plug, and this is your secondary coagulation. And so this is a clot or a thrombus. Again, I'm not going to go through the cascade of factor activation at this stage. So you've got a clot. It's fibrin protecting the damaged endothelial wall, trapping red blood cells, lots of platelets there. Then actin and myosin and the platelets contract and make the plug slightly smaller. And then as the tissue underneath is repaired, then plasmin produced by the liver from plasminogen breaks down the clot by fibrinolysis. Okay, so I'm talking about red blood cells, platelets. I'm going to move on to the immune system. Now, the immune system is spectacularly complex. And I'll talk about natural killer cell literature developing over the last few years. Just when you think you've got a handle on it, the goalposts move. So I want you to get a grasp on some of the basics. And the way that we like to classify things is useful, but it's not absolute. So innate immunity is your immediate response to infection. It's not specific, and it doesn't require memory. You can't, if you have an infection, you can't wait 2 or 3 days for the cell-mediated system to kick in. You'll be dead by then. So you have to have a very, very fast-acting system. It's primitive because a very similar process is conserved throughout evolutionary time. So it's an effective system, but it's not particularly smart, and it doesn't have a memory, but it can prime the adaptive immune system. And some of the cells overlap. So these divisions are not absolute. It's just a convenient way of explaining it. There are a number of components to innate immunity. I mean, the best one, of course, is the skin. It's a physical barrier. And there are a number of chemicals. You've got defensins produced in the gut by damaged keratinocytes. So in response to infection, there are innate molecules released which have antibacterial activity. I haven't talked about toll-like receptors here. Toll-like receptors provoke inflammation in response to exposure to bacterial coat proteins, viruses, double-stranded RNAs, and things of that nature, and they create inflammation. So again, inflammation is an innate response to infection. Claire will talk about the complement system, where you make invading material more antigenic. What I want to focus on is some of the effector cells of innate immunity. And remember, these are not absolute. This is fluid. But I think it's a useful way to get a handle on this. So you've got tissue-resident cells, like mast cells are important, macrophages are important. Neutrophils are rapidly recruited to the site of infection. They're phagocytic, and so they will engulf invading organisms. Eosinophils involved in fighting helminth worm infections. Natural killer cells, again, kind of a lot of themselves. They're lymphocytes, but they don't express B or T cell receptor. Well, some do. Some express T cell receptors. So they're T natural killer cells, which is distinct from natural killer cells. And natural killer cells, they will recognize cancer cells and viral infected cells as well, which I might talk about a little bit if we have time. So I want to keep an eye on time. I haven't drone on for too long. We looked at neutrophils before, but if you recognize anything in a blood film, recognize a neutrophil. So they're also called polymorphonuclear cells because of the multi-lobe nucleus. Normally 3 lobes, they could have 5 lobes. These lobe cells, sometimes students get confused with eosinophils because these do have granules. If you see an eosinophil next to a neutrophil, eosinophils are much, much darker red granules. And the eosinophils have a lobe nucleus, which in a moment it's not quite so clearly pinched off as it is here. And as we spoke about in an earlier lecture, I think it was connective tissue, they're full of granules, and these granules, of course, allow them to engulf and destroy invading organisms. So neutrophils mature in the bone marrow from a myeloblast, a very early myeloid cell, through promyelocytes to metamyelocytes. Again, looks a bit like a monocyte, but of course you never see these in peripheral circulation. The only time you see these in peripheral circulation is if they become arrested in a leukemia, and the sheer force of numbers expels them from the bone marrow. So the latest stage, and probably quite recognizable, is a band cell, and then the band cell is the precursor of a neutrophil, and at this stage it's released into the circulation. And this process is under control of granulocyte colony stimulating factor, which also has other roles as well in activating neutrophils. So if you look here, it's probably a metamyelocyte there with a lobe nucleus, myelocyte or promyelocyte, and then a band cell. And that's the cell which is the step before the formation of the mature neutrophil. So again, if you recognize anything in a bone marrow smear rather than a peripheral blood smear, you should recognize a band cell, a very characteristic nucleus which you're going to pinch off and adopt the conformation characteristic of neutrophils. So neutrophils full of granules. You've got your eosinophilic granules, and these contain a range of enzymes or molecules involved in killing invading microbes, defensins, lysozymes, and other molecules involved in modulating the immune system. I don't want to go into too much depth because all immune cells also produce their own cytokines which can either recruit more immune cells or they can suppress immune reactions, and I'm not going to go into that in any depth. One of the reasons being I don't really understand it fully myself, so I wouldn't expect you to either. So you think about a case of infection. Well, if there's a wounding itself will cause inflammation, stimulate histamines, which will make blood vessels leaky. They'll produce neutrophil chemotactic factors, and so at the site of wounding and infection, neutrophils will rapidly be recruited from the peripheral circulation. I think a useful thing to remember, if you think of a blood cell in circulation, other than red blood cells, the function is of course when they make it to the site of activity. So neutrophils are circulating until they're summoned to the site of infection. So again, we've got this diapedesis, the neutrophils leave these leaky blood vessels in response to the chemotactic factors released from the site of the wound, and then they engulf invading bacteria. So you can't hang around waiting for the adaptive immune system to kick in. It should be dead. So these will mop up the vast majority of infections. Anything which breaches the barrier, which makes it past the first line of defense and molecules, will rapidly be mopped up by the innate immune system. So it's first line of defense. So other circulating cells, and again, I think we're going in order of frequency. Neutrophils, most common circulating nucleated cells, your red blood cells, are the most numerous cells in a blood smear. Monocytes, characteristic kidney-shaped nucleus. Now, if you're looking at a blood smear, if you take any individual cell, it might not necessarily reflect every aspect of the histology of that cell type. But as I've told you before, one of the beauties of having access to the digital slides, you can look around and you can find cells which encapsulate those properties. And if you take a blood smear, have a look around, you'll find these cells which are slightly larger than neutrophils. It says 12 to 15 micron diameter. My feeling is more like 15 to 20 microns. They're agranulocytes. They do have granules, but they're very, very fine granules, and they've got the indented or kidney-shaped nucleus. So monocytes are easy to find. So could you tell me, what do monocytes differentiate into in tissues? Excellent. Is anyone feeling slightly regretful of that statement? No? Right, okay. Well, I guess, functionally, in the right ballpark. Okay, so reticular endothelial system. This is actually quite an archaic term. Again, what it really describes is a distributed system of macrophages. The monocytes are, as far as I'm aware, the precursor of all macrophages. Macrophages are called different things in different places. Osteoclasts are the giant cells, and they're by sort of conglomerations of monocytic precursors. So osteoclasts derive from macrophages. They break bone down. Kupffer cells, they are the macrophages of the liver. Dust cells, macrophages of the lung. And the whole in the skin, you see tissue macrophages. You've got the microglia in the central nervous system as well. These are all derivatives of monocytes. And so the lymph nodes, the liver, the spleen, they provide a distributed reticular endothelial system. They're filtering lymph in the case of lymph nodes, blood in the case of spleen, and so as they're encountering defective cells, infection, the macrophages there can deal with it. Eosinophils, again, very easy cells to spot. The granulocytes, the granules, very eosinophilic. They do have a kind of kidney-shaped nucleus, but if you see an eosinophil next to a monocyte or next to a neutrophil, always look at things in context. Very, very clearly different. Again, surrounded by some red blood cells here. These are much less common, but you will see them in blood smears. Quite a shortish lifespan, but they've fairly rapid turnover. You see them in various mucosa. You don't see them in lung unless there's a chronic inflammatory condition such as asthma. But you see they're quite common in the intestinal lining, although saying that, I don't recall coming across that many looking at our histological slides. They contain a number of components in the granules which are involved in immune responses and in killing as well, because the main function of eosinophils is probably to deal with parasitic helminth infections. They may also be a role, they are mildly phagocytic. They may also have a role in destroying antigen-antibody complexes as well. But if there's a helminth infection, eosinophil counts go up. So you see a blood film full of eosinophils, you might suspect a helminth infection. Basophils are very similar to mast cells, functionally very similar as well. Initially thought to be of the same origin, but I showed you earlier on they actually have a slightly different derivation. But to all intents and purposes, they're a circulating version of mast cells. There's some evidence that they actually migrate to sites where mast cells are active to complement their function. Do very similar things. They've got histamines, which make vessels leaky. They've got heparin, prevents blood clotting agents, and they undoubtedly have an innate role, but the best characterized function of basophils is their association with allergic reactions. Wherever you've got type 1 hypersensitive reactions, you'll see a lot of basophils. So when you get re-exposed to an allergenic challenge, you get very rapid degranulation, histamines, heparin produced, and you get local swelling, edema, hives, the things we talked about in earlier lectures. So again, basophils, here is in circulation. You don't see mast cells in circulation, but basophils, very, very similar. So these cells, in part, constitute a big chunk of the innate immune system. The adaptive system can be, for purposes of teaching, classification divided broadly into two systems, but of course there's crosstalk between these systems. So you've got humoral immunity, which means it uses the humors, the fluids of the body, and these are secretory molecules, antibodies produced by B cells. And then the main effector of cell-mediated immunity is the T lymphocyte. Again, there is crosstalk with B cells and indeed the innate immune system, which can prime subsequent reactions or responses to immune challenge. So all lymphocytes originate the bone marrow. B cells produce antibodies. They undergo somatic recombination in the bone marrow, which I'm going to talk a little bit about. T cells originate in the bone marrow, but they migrate to the thymus. Say in younger individuals, they'll mature in the spleen. In adults, and they will differentiate in the T helper and T. That should actually be T cytotoxic cells there. To change that slide, and natural killer cells as well. And again, there are at least two different kinds of natural killer cells. You've got some which express T cell receptors, so they're T natural killers. And then you've got other natural killer cells, which they're cytotoxic. They kill cells, but not in MHC context. They're not making decisions about self or non-self. If they see a host cell which is aberrant in some way, for example, it's become transformed or it's virally infected, they recognize a change in cell surface markers or interferons, for example, released by a virally infected cell, and they will kill that cell, but not in MHC context. I'm not going to go into MHC context in any great detail, other than to illustrate how T cytotoxic cells function. Okay, so key thing about lymphocytes, they've got receptors for non-self, so B cells and T cells have got receptors which recognize foreign antigens, particularly B cells, receptors for foreign antigens. T cells will also recognize cells, so T cells will only kill a cell which is presented in context. So it's an extra control to mitigate autoreactivity, which I'll talk about a little bit. So they're very, very selective in the cells that they kill. Natural killer cells, a bit less discriminatory. If there's a cell which is bound to an antibody, they'll recognize the constant region of the antibody and kill it. And so they'll also kill tumor cells to a greater or lesser extent, and virally infected cells. Again, you want to kill a virally infected cell quickly before the virus replicates, takes over the organism, and kills you before the adaptive system kicks in. Now, I'm going to spend a couple of minutes explaining this. Now, I've asterisked this because I promise not to examine you on it. But I think it's important to think about where diversity comes from in the immune system. And I apologize to anyone who knows this already. The other reason I'm going to talk about this is I spent 6 years of my life sequencing these things in the days before we had machines that sequence things. So if I was miserable, I think I can make you uncomfortable for 5 minutes as well. And if you think about it, where does all this diversity come from? How can B cells recognize any challenge that they're exposed to? And an antibody is created from two chains. So you've got 2 copies of a heavy chain, 2 copies of a light chain. That's 2 genes. How can those 2 genes produce a polypeptide product which could recognize any challenge? And they do it by a process of somatic recombination. And they chop up the DNA. So whenever somebody says every single cell in the body has their, your full complement of DNA, that's not true. There are 2 exceptions to it. Can anyone think what the exceptions are to cells that don't have full complement of DNA? Sorry? So we've got B cells, T cells, and red blood cells, of course, which are anucleate. And so B cells and T cells chop up the DNA, and they lose part of the immunoglobulin genes to create a hypervariable region. And the process is actually quite straightforward. So on the germline, you've got an array of somewhere, I mean, the numbers will vary with haplotypes, so different individuals have different numbers of variable segments. We have a run of 100, 200 blocks which create a variable segment, 26, 27 diversity, 6 to 9 blocks in the junctional region attached to a constant region. And what happens is, through a process of somatic recombination, is that one V segment is selected, one D segment is selected, one J segment is selected, and they're brought together. So if you do the math, if you multiply 123 by 27 by 6, you get something about 20,000. Obviously, you can make many more than 20,000 antibodies. And what happens is exonucleases nibble away at these blocks and random nucleotides are inserted. And once this has rearranged, it can rearrange again. So an antibody can be honed to increase its specificity for an antigen. And this means that 2 genes can produce antibodies where they can recognize just about any challenge the cell would ever meet. So what happens in a B cell precursor, it'll shuffle up its DNA. In the bone marrow, you'll have millions of B cells all expressing different immunoglobulins. They will then be selected. You want to select autoreactive clones, so they will be deleted, die by apoptosis in the bone marrow, and then B cells expressing different receptors will be released into the bloodstream. Does that make sense? Does that demystify that for everyone? Excellent. So these nodding happy faces. Wonderful. So say, I want to examine you on this, but I think it's a useful principle to understand, because it's the same for T cells as well. This is where diversity comes from. So in this rather facile cartoon, we've got the B cells here. So at this stage here, I guess, this is where the recombination is taking place. These B cells all come from a common precursor, but they've all shuffled the DNA by somatic recombination. So each one of these will go on to express a different immunoglobulin. The important thing is to remember, of course, there won't be one on the surface, there will be thousands, but each will be the same. So these little shapes are trying to represent each B cell is unique. It will recognize a different antigen. These then go into the circulation. In the lymph nodes, if they recognize an antigen, that cell will become activated and it will expand, and it might undergo further mutation as well to hone that interaction. I think it's shown in the next slide. So if the B cell gets activated by recognizing an antigen, a couple of things can happen. One, by interaction with the T helper cell, and again, this is where this crosstalk between the systems, it will promote the class switching activations. You get the division, the proliferation of the B cell, and rather than having a membrane-bound antibody, it'll start pumping out the antibody. So this antibody goes into the circulation, and then it will meet and precipitate macromolecules or target invading microorganisms for killing. And again, these exposed FCs will light up when they bind to an antigen and their targets for natural killer cells. But of course, you want to create a population of memory cells as well. And stepping back at plasma cells, these are long-lived. These can live for years. And if you live for long enough, eventually these will transform, and plasma cell leukemias become progressively common in aging. It's quite unpleasant. And so memory B cells, these survive for many, many years. When you're re-challenged, they can re-expand and provide a new source of antibodies quickly to the memory of a previous infection. Okay, that's the easy stuff. Now it starts getting a bit complicated. I'm going to try and simplify it. So T cells, the cell-mediated immunity is spectacularly complex. Any textbook is probably years out of date. And immunology is a very arcane field, its own technology, things are changing all the time. But some of the basic principles are worth remembering. So T cells don't express immunoglobulins or B cell receptors, they express T cell receptors. But they have a range of other co-receptors as well, because you have to have co-stimulation to activate a T cell. And one of the reasons is to minimize the chance of reacting with on killing self. So I guess a key function of T cells is recognizing self and non-self, and recognizing self in the as non-self molecules in the proper MHC context. I won't go into that in any great deal of depth. So the progenitor lymphocyte migrates to the thymus or the spleen, undergoes rearrangement of the T cell receptor genes, and then I'll upregulate either CD4 and T helper cells, CD4, and CD is cluster designation or cluster determinant. It's just a useful nomenclature system for classifying cell surface molecules, and it's widely used in hematopoiesis. So CD8 is expressed in cytotoxic cells, CD4 on helper cells. So CD4 positive cells, in response to an immune challenge, they will, as mentioned earlier on, they're involved in memory B cell formation, for example. They'll also activate macrophages, where CD8 cells, in the right context, will introduce apoptosis. So in this diagram here, I guess I was trying to show is you've got an immature precursor, migrates to the thymus, it matures there, upregulates its T cell receptors, and also upregulate multiple copies of either CD8 or CD4, and then they're selected for dual positivity. And of course, anything which is cells which don't react with anything are deleted. Cells which are strongly autoreactive are deleted as well, and you end up selecting a population of cells which are mildly autoreactive, so they will recognize antigens presented by self cells in MHC context. Now, don't worry about that too much. Claire will explain that a bit more clearly next term. Sorry, after the Easter holidays. So basically, two populations of cells expressing the T cell receptors. Again, these have shuffled up the genomes again to provide that huge repertoire for for recognizing antigenic determinants, and then you've got your co-stimulatory molecules, and other cell surface markers characteristic of cytotoxic or helper cells. So, if a macrophage has ingested an antigen, it will ingest it, it'll chop it up, and express it on the cell surface, and it's in the sort of surrounded by your major histocompatibility proteins. So, a T cytotoxic cell will recognize this foreign antigen expressed on the surface of a cell, and then it'll kill it. Actually, this peroxide comes from the macrophage. What it does is it'll punch holes into the membrane of the macrophage and then fill it with proteases, which will lead to the death of the cell. And then T helper cells, they can do a couple of things. They can produce cytokines, which I guess will either recruit more T cells or suppress the reaction. And they're also involved in the establishment of memory T cells as well. So when you're re-challenged again, you can quite quickly mount a cell-mediated response to this. So I think I will go through this actually. I think it summarizes quite a few key features. So natural killer cells, they destroy damaged cells. They destroy self cells, part of the innate immune system. So if a cell is infected by a virus, for example, it'll pump out, the cell surface molecules will change, it'll produce interferons, and that'll recruit natural killer cells. Tumor cells will downregulate their MHC molecules to try and evade detection, and natural killer cells can recognize this and kill them. And I'm sure you've heard recently about immunotherapy. I mean, actually, immunotherapy has been around for years and years and years. When I did my PhD in hematology back in the dark ages, and it was known that if you gave somebody an autologous blood cell transplant, so you apheresis, you collect the stem cells, you mobilize the stem cells with GM-CSF, you collect them, then you ablate the bone marrow, you kill everything, you kill the niches that contain the tumor cells, you put their own bone marrow back in, which either repopulates the bone marrow or allows residual hematopoiesis to recover. There was a graft versus leukemia effect. So tumor cells are antigenic, and so one of the mechanisms, they downregulate their MHC genes. Natural killer cells can recognize that to an extent, and a number of different things you can do. Like interferons can prime the immune system. You can expose, you can apheresis populations of T cells, train them to recognize damaged cells, viral cells, and so viral infected cells or tumor cells, or you can modulate a response already exists. So a number of things you can do to tweak the immune system, which will allow it to kill cells which had previously escaped surveillance. And of course, this immunotherapy is made into the news, I think, because a couple of companies now are producing treatment regimes where they take a patient's cell, they expose them to a tumor-associated protein, stick it back in again, and have enhanced tumor killing. So, and largely facilitated via natural killer cells. So B lymphocytes produce antibodies, humoral production of humoral release of factors which kill, inactivate, precipitate macromolecules or pathogens. T helper cells, and again these are the populations that get impacted in HIV. A number of different, they help B cells turn into plasma cells, produce immunoglobulins, they activate macrophages, and they're also involved in modulating cytotoxic responses. CD8 cells, they recognize foreign antigens expressed in MHC1 context. They won't kill a foreign cell, like a cell has to be engulfed by a macrophage, so they have the self and non-self antigens. And they do it by punching holes in the cell, putting in granzymes containing proteases. I've bracketed the last three. You'll hear about T regulatory cells, the responses that involve intolerance. There are natural killer T cells as well. They've got a range of different functions. They're different from these natural killer cells. They come from the same origin, but they express T cell receptors. Nobody really knows why, but they do produce a lot of cytokines. I've also put in gamma delta lymphocytes. B cells go heavy in light chains. Most T cells rearrange the alpha and beta chains, and so most T cell receptors are encoded by alpha and beta chains. In the periphery, in skin and mucosa, you've got gamma delta cells. They don't recirculate, and they're an innate-like counterpart of the T cell system. Okay, so I'm not going to read through the learning outcomes. That's us done for today. So one more lecture after Easter, so enjoy your break. And I'll see you in a couple of weeks' time. Thank you.
Thank you.